Nitrogen fertiliser prices are getting out of hand. So farmers are looking for alternative ways to supply their crop or their nitrogen needs. And so here I have 5 ways we can reduce our nitrogen inputs, 3 of which we're actually making our own nitrogen inputs, and the other 2 are minimising the nitrogen loss when we do apply nitrogen. Real quick, my name's Till Simmons. I help growers transition from conventional way of farming to regenerative way of farming using practices like these to reduce our nitrogen inputs and many other things. If you want to work with us to improve your farm's soil health and all these other things, make sure to reach out to us. I have a link to our website below so you can check that out. We have a building soil health in 90-day program at the moment. So if you're keen to build some soil health and reduce your nitrogen inputs, then get in contact with us. Cool, so there's 5 ways that I have identified in reducing our nitrogen inputs. They are firstly growing legumes, using cover crops, microbes, converting all our nitrogen to amino acids, and using humic substances. So I'll explain each of these, starting with legumes. So legumes are able to have this symbiotic relationship with rhizobia bacteria, and basically this is a relationship where the bacteria fix their own nitrogen in the nodules of the legumes. And so if this is our legume plant here, basically the legume can take in these rhizomium bacteria to form these little nodules on the plant roots. In these nodules, there's heaps of these bacteria, and they start fixing nitrogen for the plant. And so the plant will give the bacteria food, sugars and whatnot, and make an environment nice for them to grow. And in exchange, the bacteria will supply the plant nitrogen. So instead of growing, say, a cereal crop, we could potentially grow a legume crop. So chickpeas, there's soybeans, there's lupins, any of these. Now they do tend to be lower yielding, and so this will depend on your farm and I guess how bad nitrogen fertilizer prices get. But that's one alternative. With this, we shouldn't need to apply any nitrogen fertilizer, as the rhizomium bacteria should supply the plant 100% of its nitrogen requirements. Now, with this, there's a big caveat. We need the legumes to actually be inoculated by these bacteria, and so they need to nodulate. Now, there's a couple of requirements we need for this to happen. Firstly, there actually has to be rhizomium bacteria in the soil or around the seed to start with. And so if there's none there, they simply won't form any nodules. So what we can do to overcome that is, firstly, we can apply the bacteria to the seed coat. So when we get seeds, we make sure we apply a product which contains the bacteria. And there's a lot actually we can go into on that, but inoculating the seeds, we can do that. The other part is that legumes won't nodulate if there's already nitrogen in the soil. So if there's a bit of nitrogen in the soil, the plant, it's more efficient for the plant to absorb that nitrogen, which is in the soil, than to form these symbiotic relationships with the bacteria, because it's quite expensive for the plant to give up all these carbohydrates to the bacteria. And so if there's free nitrogen in the soil, they'd rather pick that up. It's more energy efficient for them. And so what we need to do is make sure there's very little nitrogen in the soil, and we can do that with a cover crop. Now, I'm pretty sure that when we do apply nitrogen fertilizer, if it's inorganic, it can actually reduce nodulation as well. So applying high amounts of inorganic fertilizer into the future will reduce the rate of nitrogen fixation for legumes. But if we have a slow release form of fertilizer, such as decaying organic matter, my understanding is that that doesn't affect nodulation as much and the rate of nitrogen fixation. So there you go. The first one, growing a legume crop, because we don't need to be applying nitrogen with that. Another one with this is that we can actually grow legumes as companion crops. And so I've seen a couple of farmers, they sow their wheat into standing clover. And so the clover, say it's like this, and it has all its nodules on the roots. The clover will be fixing its own nitrogen. And then so we plant our wheat next to that, and roots come down. What can happen, although the cereal itself is not fixing nitrogen, if we have really robust biology in our soil, what can happen is that mycorrhizal fungi can colonize our legumes and then come over and colonize our cereals. And so in small amounts, the nitrogen fixed by the legume will actually get transferred to our cereals. Now, I'm not exactly sure how much nitrogen this contributes, but I know that they do contribute some. And so this is a way that we can experiment with reducing our nitrogen inputs. Cool, so there's two there for planting legumes. Firstly, having the legume as our cash crop, and the other one is planting it as a companion crop to supply our other crop with nitrogen. But we need mycorrhizal fungi with that as well. So we need to inoculate it and make sure all the conditions are for mycorrhizal fungi to thrive. And we'll talk about that when we get to microbes. So for our legumes to really function properly, we need firstly for them to be inoculated with rhizomium bacteria. We also need to make sure that our soils have enough gas exchange for nitrogen gas to go into our soils. So we want to make sure our soils have a high gas exchange, which we can only do that with improved structure. And so when we improve our structure, we get increased gas exchange come in, and the gas can then enter into the nodules. We also need very little amounts of nitrogen in our soil, which I touched on before. And finally, the bacteria in the nodules require molybdenum, which is a plant nutrient, in order to fix the nitrogen. So if there's no molybdenum, MO, in our soils, then these bacteria will not fix nitrogen. Well, they will in very small amounts. I believe they can sub out iron, but it's very small and not as effective. So we need to make sure we have enough molybdenum, and we can do that. We could potentially do that by applying it in the seed coat with the inoculation. So if we make up a solution with the bacteria, maybe have some chemical substances and a source of carbon source with that, and then we also add our molybdenum, that would be a really good mix to get these bacteria really going. So molybdenum is very important, and iron should have enough in the soil as it is. But a lot of soils are deficient in molybdenum because the biology is just not there. Next, cover crops. So cover crops for this example work in a way that they're going to pull up all the nitrogen essentially out of the ground and then move that into amino acids. And so if there's nitrogen in our soil that would otherwise be leached, so we're going to lose that nitrogen anyways. If we're going to lose that nitrogen, we might as well turn that into plant biomass and amino acids. The main function of nitrogen in our plants is a building block for these things called amino acids, which in turn, these things are building blocks for proteins. So essentially we need nitrogen for building proteins. And then proteins are used in everything in the plant. So making enzymes and structures and all these different things we need nitrogen for. And so that is why nitrogen is the probably most important plant nutrient and is required in the largest amounts. So the cover crop will pick up all of that free nitrogen in the ground that would otherwise get leached out to build amino acids, and we store that nitrogen in amino acids in our cover crops. There's all these other benefits that we get from cover crops, including increasing our microbial community. With the root extracts, we increase the organic matter, we increase infiltration as roots go down. There's all these benefits that we get from cover crops, and nitrogen is just one of them. And then there's other things that will supply our cover crop nitrogen, and we'll get to that in a second. But what we need to do now is we need to get the nitrogen in our cover crop back into the ground so they can supply our next crop with nitrogen. And so what we're going to do with this, we're going to either terminate it with like a crimper or graze it so that we can get the biomass in the cover crop onto the soil so they can start breaking down. And so when we think about it, all through our plant is nitrogen and protein and amino acids. When our plant is now on the ground, it can come in contact with soil which contains our microbes. And so they'll start breaking down the biomass into its individual parts. So it'll be releasing amino acids, it'll be releasing sugars and nitrates and ammonia, all these building blocks of the plant back into the soil. So as a general rule of thumb, cover crops will contain about 3% nitrogen or just plants in general, about 3% nitrogen, dry weight. Now, half of that will be available, so half, which is 1.5%, will be available 1 year after that cover crop is terminated. Now, if we have really robust biology in our soil, that will happen sooner. If our soils are pretty dead, that might happen a lot further out. And the reason why we only get half is that the microbes in our soil will eat a lot of this biomass and put it into their own bodies. And so the nitrogen in our cover crop then gets converted basically into microbial biomass. And from there, they'll get eaten by other microbes or even our plants, which we're going to touch on a bit later. And then when that happens, a waste product of that is a form of nitrogen that the plant can pick up. So yes, the plant can pick up amino acids whole, so whole amino acids which are from the breakdown of our cover crop, as well as amino acids released from our microbes and any other nitrogen that our microbes throw out. And so pretty much if we can grow as much biomass that we can per hectare, we'll produce more nitrogen next year and the year after and the year after that. So then really after 12 months, we have a 1.5% of the nitrogen from our cover crop released, which means we still have 1.5% of the cover crop stored in our soil. And so 12 months after that, 50% of that will then become available. And so what happens is that over time, as we increase the amount of cover crops we have, we slowly approach, I guess, the full amount that the cover crop would release in one go. As each time we have a cover crop, it adds onto the previous cover crop. But this is a free way to get a nitrogen, and as well as to reduce the amount of nitrogen that's leached out of our soil. So if we can do this to produce our own nitrogen, then we won't have to apply as much nitrogen fertilizer. The best way to do this is to take a whole tissue test of just a plot and see actually what it comes back at from the labs. And so then you can estimate the amount of nitrogen credits, and then you want to monitor your plant across the growing season to make sure that they're not nitrogen deficient. Now, if we really want to get more nitrogen from our cover crop, we can increase the amount of legumes that it has. And so when we increase the amount of legumes, it's going to be increasing the amount of nitrogen fixation from our first point into the cover crop. And so instead of having 3%, it can be 4% of the biomass, and we'll be producing even more biomass. So the third way we can reduce nitrogen inputs on our farm is by increasing the microbial activity and populations within our soil. And so there's three ways that this can happen. The first way is the mycorrhizal fungi. Firstly, by taking nitrogen from our legumes, and secondly, from actually helping the plant absorb nitrogen in our soil. So they can help our plants absorb nitrogen. Mycorrhizal fungi, for those that don't know, it's a fungi that attaches to our plant roots and basically extends out the plant roots. And so this network of fungi really helps the plants to increase its area that it can absorb nutrients from. The mycorrhizal fungi will exchange these nutrients with the plant for sugars, and that is how the relationship works. And it can also connect them to other plants, supply different hormones and all these things to the plant. So increasing our mycorrhizal fungi in our soil, we can do that with inoculating, but also making sure that the environment for our soil is good for mycorrhizal fungi to thrive. And so I have another video which explains the three things we need to improve our soil health. But in short, it is making sure the microbes are there. We need to feed them, so root exudates, and the environment to make sure that they're nice and comfortable. So that's making sure that the soil is nice and aerated. And then finally with that, we've got to make sure we're doing things that won't kill our microbes. So we're not tilling up our soil, we're not applying really harsh inorganic fertilizer, and the aim of this is to reduce that. So I guess over time, naturally, we will be increasing mycorrhizal fungi. Another way we can make our own nitrogen is with azotobacter, which is a free-living bacteria in our soil, which makes its own nitrogen. So just like the bacteria in our legume nodes. These bacteria are free living within our soil and can do all of that by themselves. Now, they're not as efficient, and they only produce about 20 kilos of nitrogen per hectare, so not much at all, but something. And when we have that in combination with all these other things, then that helps. So there's that type of bacteria too. And finally, a process that I've learned from John Kempf is the rhizophagy cycle. Now, this is a pretty new discovery in the ag space, and essentially what happens is on the growing root tips, the root basically absorbs bacteria, rips off their skin, eats it, and then throws the bacteria out. Now, there's a bunch of different things that actually happens, but if you want a quick overview, that's basically what happens. And so in the skin of the bacteria, there are amino acids and amino sugars and nutrients and minerals and all these things that the plant needs to grow. So basically what happens is that the plant can absorb these nutrients from bacteria growing in our soil and then use those nutrients to grow. And so this requires us to have really robust biology in our soil, which is the more biology we have, the better our plants can use this cycle and grow. What inhibits this cycle is the use of lots of inorganic fertilizers and lots of tillage. So when we till up our soil, releases or shows the bacteria to the sun, and they don't like that and die. And so when we are using no-till, we're covering our soil, we're using carbon-based amendments, then we're able to increase the amount of bacteria and microbes in our soil. And so that's another way. Dr. James White, which is the lead researcher on this, he suggests that the plant can get 100% of their nutritional requirements from this process. And so it's a revolutionary process that we've just kind of found, but definitely something to look into and see if we can start tapping into that cycle. And so for that, we need to make sure we have our bacteria and fungi. And so we can do that by inoculating the seed coating. The best way to do that isn't just with one strain of bacteria. Like a lot of products, they go, oh, we've got this one strain of bacteria, it's really good. What we want to do is apply as much stuff as possible, because we still don't exactly know how the soil works. And so if we're just going, oh well, let's apply one bacteria, it doesn't really give the full picture of the system. And so it's best to just go, yeah, we don't really know exactly what's going on, so let's apply everything, and the plant and the microbes can figure it out between themselves. And then with bacteria, there's the rhizobium bacteria as well, which are in the legumes. So that covers our first three, which are making our own nitrogen, mostly using bacteria and microbes. Take the nitrogen from the sky and put it into a form which the plant can pick up. The last two are different things we can do to our fertilizer applications to make sure that we're not losing as much nitrogen when we apply it. So there's a couple of reports that suggest that up to 60% of our nitrogen is lost. And so imagine paying for something and only getting 40% of what you actually paid for. So that can be in the form of leaching, so you lose a lot of nitrogen from leaching. Volatilization, so it's pretty much gases off to the atmosphere. And so one way that we can fix this problem is that when we apply our nitrogen, we want to convert it straight into amino acids straight away, as soon as possible. And so this requires almost a similar process to our cover crops and what we talked about there. But basically we want to make sure that when we apply our nitrogen fertilizer, the microbes in our soil have enough ingredients to then take that nitrogen and straight away combine it to themselves in their biomass. And so full credit to John Kempf, this is where I learned this from. He has a program which you can check out on their site, but essentially they recommend to have a carbon to nitrogen to sulfur application of 300 to 10 to 1. And so the carbon gives the microbes energy to actually do this process. Nitrogen is the nitrogen that they'll take up for their own bodies. And then sulfur. Sulfur is really important for the building of amino acids as well. And so if we don't have sulfur, we get incomplete proteins. And so that's not very good. And the microbes are limited by basically almost this ratio. So when we have all of this in a fertilizer application with microbes and biostimulants and stuff to get our microbes kicking, then we can effectively convert all of that nitrogen straight into a stable form, which is in the microbes biomass. Now there's one more thing that we need to add to that, and that's molybdenum. I don't think there's a specific ratio relative to these that you need to add, just enough to get it sufficient in your soil. And that serves a similar function to the molybdenum used in our legumes. It helps our microbes fix the nitrogen that they're given into amino acids. So when we use this, John Kempf estimates that a lot of his growers are able to reduce their nitrogen inputs by over 40%, like across the board. So if I was you, I wouldn't go straight into that potentially. Just in case, I would maybe start the same application of nitrogen, still applying all of these, and then do a strip of say 40% reduction, and then see how that trial plot goes, and then slowly over time see where we can take what's the bare minimum of nitrogen we can apply without taking too many risks in completely cutting it out. A lot of his growers, yeah, they're able to reduce it by, I think it was at the minimum 40% across the board. And the final one is combining our nitrogen to humic substances. And so humic substances have a lot of cation and anion exchange points on the molecule, and so it's able to hold on to all these different forms of nitrogen. So we have nitrate, which is an anion, and we have ammonia, which is a cation, and so it has a positive charge. Humic substances have both negative and positive sites, so they can hold on to both of these. And so when we combine humic substances to our nitrogen applications, it holds on to our nitrogen, and when it goes into our soil, it's not going to leach away. So one study that I read suggests that they can decrease the amount of nitrogen leached by 25%. And so when we think about that, what's 25% of 60? It's a fair decent chunk. And so we can effectively, by adding humic substances, we can then reduce our nitrogen inputs. Now when they're held onto our humic substances, it acts more as a slow release into our soil, and so over time it's going to slowly release. So there you have it. There are some of the 5 things that I have learned about reducing our nitrogen inputs. There's a lot of research coming out about all of this, but it seems to all come back towards microbes and building our biology in our soil. We can do that with increasing our soil health and all the things that come with that. So if you are interested in trialing something this on your farm or improving your soil health, reach out to us. We have a program to help farmers do that. A very easy way to help growers increase their soil health so they can reap all these benefits. Also, well, if you enjoyed that, make sure to subscribe. And if you know anyone that would benefit a lot from reducing their fertilizer inputs, and I know there are a lot of farmers at the moment that would appreciate that, would you mind sharing this video with them and the rest of our videos? I'm really trying to build this channel out, so I would be forever grateful if you can share it around. Awesome. Well, thanks very much for watching. Cheers.